This is Volodymyr Krout, and the crash course for agronomists simply about microbes. It's time to discuss a very important and interesting issue that worries everyone who cares about their plants, no matter if it is a large field or a small garden. Today, we are going to talk about diseases. In general, pathogens are divided into four large groups. The first one is NECROTROPHS, the parasites that feed on the dead tissues of their host plants. They include causative agents of Fusarium and Septoria. The second group, BIOTROPHS, are microorganisms that feed on the living tissues. These include, for example, Puccinia, which causes rust, or Phytophthora. Interestingly, some pathogens can first behave as BIOTROPHS, and when the depleted plant dies, they switch to the necrotrophic type of feeding. The division by feeding type depends on what happens faster, the death of plant cells or the development of a pathogen. The third group, endoparasites, are pathogens that develop inside the tissues of the host plant. These include viruses, mycoplasmas, a large number of pathogenic bacteria and some fungi. The fourth group is ectoparasites, for example, powdery mildew, dodder and broomrape that vegetate on the surface of plants. Do you know that as long as a plant is healthy and strong, some pathogens can peacefully coexist with it? But due to stress or mechanical damage, they are activated and cause diseases. This phenomenon occurs in human bodies too. You would have heard about opportunistic pathogens that do no harm while a person is healthy. To sort out the issue of diseases, one should understand three important terms: pathogenicity, virulence, and aggressiveness. Pathogenicity is the fundamental ability of one organism to cause disease and harm another one, a plant or an animal. Virulence is the ability of a pathogen to infect a particular species or even variety of plant. And aggressiveness is the ability of a pathogen to quickly overcome the host's protective barriers, multiply in its tissues, be transmitted to other organisms, and survive in adverse conditions. For example, Sclerotinia sclerotiorum is a highly aggressive pathogen because it can develop quickly on affected plants and is well preserved in the soil due to sclerotia. But fortunately, it's not highly virulent, and it usually affects sunflower, soy and rapeseed in the field. And microorganisms that use the secretion of living and, most importantly, healthy plants have learned to protect their hosts from pathogens. To do this, they synthesize antibiotics and non-specific metabolites and secrete polysaccharides that create additional protective layers for plants. Besides, do not forget that each parasite may have its own hyperparasite. Let's take an example of how the hyperparasitic fungus Coniothyrium minitans helps fight the white rot pathogens Sclerotinia sclerotiorum and Sclerotinia minor. Sclerotinia is known to create very strong and tenacious forms of dormancy, sclerotia. Outside, they are covered in modified mycelia hyphae, which form a very hard shell. Inside, sclerotia contains very little moisture, but an extremely large reserve of nutrients. What does Coniothyrium minitans do? Meeting sclerotia in the soil, it first softens the shells, then penetrates inside and feeds on its mycelium. To do this, Coniothyrium minitans forms enzymes, chitinase and beta-1.3 glucanase. Then its hyphae penetrate into the sclerotia, where they cause cytoplasm lysis, that is, complete destruction of the internal contents of cells. In addition, Coniothyrium minitans forms very interesting antibiotics, macrosphalantes. They inhibit the development of sclerotia mycelium. So we see that in nature, parasitism is not a bad phenomenon, but only one of the methods of biocontrol of certain species. And in the hands of an educated agronomist, this is a convenient tool that would allow him to influence biological processes in agrocinosis. In the next video, we will tell you more about pathogenicity factors, as well as other methods of antagonism, in particular how bacteria can fight pathogens. Subscribe to our channel, ask questions in the comments and share this video with your friends. See you soon!